Can you hear me? So again, good afternoon and welcome to your class in ATC 111. So for our next lesson, we're going to talk about gene coding. So last time we talked about um, DNA extraction. So have you done your DNA extraction already, Teresa? Humana ka sa DNA.
Wala pa, ma'am.
So hope you can try no next whenever you're ready. So since we will be talking about genes at this point, we are still under module 2 and we're running out of time kasi 1 month to go na lang is our finals na. So as to describe genes, the early ideas on explaining how genes work came from actually studying human diseases by in 1902 by Archibald Garrod, and he studied alkapotonuria, and he recognized that disease is inherited via recessive allele. If we're going to review our previous topic, when we say recessive, yung hindi dominant, yung nandyan siyang characteristic mo pero hindi siya lumalabas. So like for example, Teresa, anong specific characteristic ng mama or papa mo na feeling mo na mana mo sa kanila? Teresa.
Siguro ma'am sa mama ko yung pagiging generous, ganun.
Okay, so medyo mahina ang signal mo. So okay lang. So siguro you mean generous, no? So that could be, and also if you notice na meron ding merong characteristics or attitudes yung parents mo na wala sa'yo, it doesn't mean na wala na sa gene mo. So although it's not showing because it's not dominant, it's recessive, so it's still in your genes, no? And he proposed that patients with the disease lack a particular enzyme, and this idea is connected genes to enzymes. So also, an evidence for the function of genes came from studying fungus by George Beadle and Edward Tatum in 1941. They studied the Neurospora crassa, and they used X-rays to damage the DNA in the cells of Neurospora. And they look for cells with a new or mutant phenotype caused by the damaged DNA. And then Beadle and Tatum look for fungal cells lacking specific enzyme. And the enzymes are required for the biochemical pathway producing the amino acid na arginine. And they identify that mutants deficient in each enzyme of the pathway. So ito yung mga ginawa nilang experiment, no? Meron silang wild type na kinuha and then sila ng X-ray to mutagenize or cause mutation, and then they grow them in a rich medium. Yung isa, no growth on minimal medium. Yung isa, growth, nag-grow siya in minimal medium with the arginine. So, it means they are arginine mutants. So, in this results, you can see na yung merong mutation na EFGH. So, ang merong plus ornithine is merong growth, no? And this 2 plus sertraline and so on. So, itong merong arginine, so merong growth sa lahat ng EFGH. So, in this one, they had conclude na itong mga argines were the one responsible, no, in the biochemical pathway for arginine. So anyways, that's just to show you how genes were discovered, no, and the functions. So then after that, the Biddle and Tatum proposed that each enzyme of the arginine pathway was encoded by mainly separate genes. So there's already an evidence na iba't ibang genes are involved in each enzyme, and therefore one gene is equivalent to one enzyme hypothesis, and today we know this as the one gene or one polypeptide hypothesis. So the central dogma of molecular biology states that when we say central dogma is just the main domain. If you become a molecular biologist, ito yung parang national anthem mo or RMC hymn mo, no? So the DNA is transcribed to RNA and then translated into protein. Sorry for the arrows. So, and when we say transcription, it is the flow of information from DNA to RNA. And when we say translation, it's the flow of information from RNA to protein. So, ang DNA, ito yung pinaka-basic. So, it will transcribe or transcript into RNA and then translated into a protein, which yung protein, yun yung um, that includes enzyme na we have known from the previous experiment that they have will will be the one responsible for a specific characteristic, no? So yung gene yung response, like kanina yung arginine, no? Yung arginine, pag wala yung arginine, so pathway, yung wala yung enzyme, so yung arginine, sa arginine pathway, so is encoded by a specific gene. So moving forward, this is just um how the eukaryotes and prokaryotes are transcripting and translating proteins. So we know that from our previous biology, we know that prokaryotes versus eukaryotes are the higher na what they call that organisms belongs to the eukaryotes because they have the what do you call that this nucleus, no,
Good
which is
afternoon, ma'am.
good afternoon, lovely. So we are talking about gene coding now, but before we in introducing the gene coding, we are still reviewing some of the basic knowledge on molecular biology. So prokaryotes compared to eukaryotes, prokaryotes are single celled um, organism like the bacteria. No, they don't have a nuclear envelope or nuclear or cell wall and so on. And so they are both of them are involved in the transcription. So again, transcription is the flow of information from DNA to RNA, and then translation is from RNA to protein. So the genetic code now is lahat lahat ng animals, whether from prokaryotes or eukaryotes, they are deciphering the genetic code required, determining how four nucleotides, it's called the ATGC or the adenine, thymine, guanine, and cytosine. And they can, with this only four nucleotides, they can encode more than 20 amino acids, no? I think we have 22 amino acids needed by the body. And Francis Crick and Sidney Brenner determined that the DNA is read in sets of 3 nucleotides for each amino acid. So sa isang amino acid, you have yung glutamine, in which glutathione is derived, is one of the essential amino acids. Now we have the valine, uh, amines. So you will know later. So each of these amino acids that are needed by our body are consist of 3 nucleotides. So it could be from the 4 nucleotides, it could be the ATG or GTC, whatever, no? So malalaman niya, and you're going to have a practical on this, no? Simple lang siya, no? If you are present on this discussion, so madali na lang sa inyo mansiran, and how maybe most of you have answered already yung ating module, ano ba yan, module 2 na practicals or laboratory. So in this are examples of the amino acids, not the methionine, I forgot the pro, histamine, arginine, aspirin, alanine, and methionine. So all amino acids change after the deletion of a base, like something C, no? And this is a one base and three base deleted. Like if you take the ACG, so magiging histogene, and then they do not change na after the third deletion. So ano itong genetic code? So we are now here. So genetic code is the set of three nucleotides that specifies a particular amino acid. Therefore, so when we say genetic code, yun yung tatlong combination ng nucleotides kanina, ATGC, no? And reading frame is the series of the nucleotides read in sets of three, or codons. So if you are not so familiar with I am talking about, so even me, no, kasi ang agriculture is a very wide science, no, and then that includes biology and molecular biology. So we are now on the domain of molecular biology. So only one reading frame is correct for encoding the correct sequence of amino acids. So, and it doesn't change, no? Tatlo lang talaga, or isang three sets ng three ang responsible for this code. So, Marshall Nirenberg identified the codons that specify each amino acid, and RNA molecules have only one nucleotide, and of specific three base sequences were used to determine the amino acid encoded by each codon. And the amino acid encoded by all 64 possible codons were determined. So imagine there were 64. So ito na yung sasagutin ninyo sa inyong practical exam. No? So these are actually lahat standard. No? Ito na lahat for all types of organism. So as I have said, you're going to make this one. So you can just copy this, no, and rewrite or copy paste. But then if ever in your exam, kasi magkakaroon pa tayo ng one final exam, so how are you going to do this? So anyway, I will give you the UCAG and the UCAG. So lahat yan UCAG. No, so and lahat din meron ditong UGAG. So, mga UCAG and UCAG. Ito lang yung UGAG, o baka nagkamali lang siya. So, this was, if I remember it right, this is the second time nga nag-discuss ako, no? The first one I discussed about the genetic code was, I guess, 5 years ago. So now this is the second time. So how you're going to do this is just you pair the letter U and this U here. So meron ka ng dalawang U, and then the third U here. Eto siya. So pag pair mo dyan, so malalaman mo na ang UUU is the genetic code ng phenylalanine na amino acid. So itong phenylalanine, leucine, isoleucine, and methionine, valine, alanine, and so on are all 63 possible 63 man to kanina na 63 possible na mga genetic codes. I mean na mga amino acids. So ano ang genetic code ng phenylalanine? So ito yung UUU and UUC, no? And then kasi you pair U here and U here and then C here, no? Kaya nagiging UUC siya. So, and also serine, meron siyang U, U, and then C, and U. Can you follow me? Ma-follow ba ninyo nga? Kaning mga first letters here are actually given na siya nga coding, no? And they are standard arrangement already. So, imuha na siya, just like when you do the sa gato yung ipang arrange ganit sa sa hybrid and monohybrid. Ang sa gato siya na tawag na. Ang sa gato siya nga box. Can you still remember? Yun yung naghimo monohybrid and
Punnett square, ma'am?
yeah, what was that again?
Punnett square?
Punnett square. So, kalimtanon kay ko, no? Sige lang kay kuha naman ko, idad-idaran na. So, Punnett square, thank you, Joubert. So, it's just like the same po siya, aning genetic code, no? Para siya kaning ano. So, i-pair ni mo po siya, no? Nakaana siya dari. And then, leucine, U, kaning U here, kaning U dari. And the letter A here. So, sa phenylalanine, UUC siya kasi ang C, where's the other C? Supposedly G siya. No? So, maligin niya siya. Dapat ko ano siya? G or C. So, let's do it again. U here, sa leucine na taha, and U, and then A here. Makita man niyo akong arrow, no? Makita niyo akong arrow. So, kaya...
Yes, ma'am.
So, kaya nasa U, U, A, and U, U, G. So, there is U, and then U, and U. And here is U and U. So, dapat, dili niya siya C, G niya siya. No? So, I will just review ha, kasi as I have told you, 5 years ago siya nako nagitudlo and lahi to siya nga kaning table. So, I will look up to although the same nagyayapon niya X1, pero I'm not using the two's table again. So ganun din ang sequence ng the rest, no? So, we have another leucine here, and it's encoded as CUU kasi C, and then U, and then here, the U. So, mas simple kasi yung ginamit ko last time. But this will be... So again, the codon consists of 3 nucleotides read in sequence here, and ACU codes for 3 and 9. And for the first letter, A is the first letter column. And the second letter, C, is in the second letter column. And so mayroon explanation dito. And the third letter, U, is in the third letter column. So each of the yung sinabi ko kanina, yung tinuro ko. And each of the mRNA codons is recognized by a corresponding anticodon sequence na tRNA molecule. So meron din siyang anti-codon, yung mga codons na ito. And many amino acids are specified by more than one codon. So, in one codon, meron siyang tatlong nucleotides ng ano nga yung U? Uracil ba yun? Cytosine, adenine, and guanine, no? So, yun yung mga nucleotides. So, tatlo-tatlo siya, pero sa phenylalanine, like example, dalawang codon siya, no? Which has three nucleotides each. So, for example, threonine is specified by 4 codons which differ only in the third nucleotide, yung ACUs, ACC, ACA, and ACG. Etong threonine, etong mga C dito, actually they have all 4 na codons, no? Tag-aapat sila, hindi lang threonine. And merong nakalagay dito AUG, methionine start, no? And meron din siyang stop. So it means nag-start dito kasi nga start, no? And here nag-stop din ang codon at this point. Kaya wala siyang kanang identified na amino acid and is as well as here. Okay, so do you have any question before we move forward? So this is already very fixed and just very simple na para siyang mathematics na it is covered by one law lang. So nakasabot mo tanan kung unsaon siya pag-pair. So you will have a laboratory exercises in which you will just copy all the table here, no? Or you copy paste. So yun lang ang laboratory exercise ninyo. But in your final exam, etong mga codons are blank already, no? So kayo muna ang mag-pair ana sa inyong exam. Okay ra? So again, in the stop codons, we have 3 stop codons, no? Nakita kanina, the UAA, the UAG, and the UGA. Ito yung tatlong stop codons. And this is used to terminate the translation. It means matatapos na ang translate ng RNA into proteins or amino acids. While the start codon, the AUG, ito yung methionine, no? Is used to signify the start of translation. And this is the remainder of the code is degenerate, meaning that some amino acids are specified by more than one codon. So, yun yung mga signals nila. So, as an overview of the gene expression, meron tayong template strand or the strand of the DNA double helix used to make the RNA. So, kanina sa first part figure natin, makikita natin yung figure ng RNA and DNA which are double helix, no? And coding strand is the strand of the DNA that is complementary to the template strand. So, it's just the strands ng DNA na which is also the same as double helix template strand. And the RNA polymerase is the enzyme that synthesizes the RNA from the DNA template. So the transcription then proceeds through ang first step ay initiation wherein the RNA polymerase identifies where to begin transcription, and the elongation wherein the RNA nucleotides are added to the 3' of the new RNA, and the termination wherein the RNA polymerase stops transcription when it counters terminators of the DNA sequence. So, and then after the transcription, the translation proceeds. So, how? First again, initiation, elongation, and termination. Kagaya pa din ng transcription. So, in the initiation, the mRNA, so kanina diba, yung RNA nagiging messenger RNA na siya and transfer RNA and ribosomes come together. So ito yung start ng translation and the elongation. The tRNAs bring the amino acids to the ribosome for incorporation na in the polypeptide. So if you remember the ribosomes in the cell parts, no, these are the one which contains or which the in where the RNAs are delivered. So termination is when ribosome encounters a stop codon, yung stop codon kanina sa genetic code, and this will release the polypeptide. And once the polypeptide is released, so again, before we proceed, these are the gene expression, no, which has participation of different types of RNA. Kanina yung messenger RNA or the mRNA. It's the one that carries the information from the DNA that encodes the proteins. Kaya messenger siya kasi meron siyang information. And the ribosomal RNA or rRNA is a structural component of the ribosome. So once na mag-enter na siya sa ribosome, meron na siyang it's for the making of proteins. No? And that lastly, the transferred RNA is called the tRNA, carries the amino acids na to the ribosome for translation. So, ito yung nabanggit na kanina here. So, we also have small nuclear RNA or snRNA involved in the processing ng pre-mRNA, messenger RNA. And the signal recognition particle, or known as the SRP, is composed of the protein and RNA and involved in directing RNA to the RER. And the microRNA or the miRNA are very small and their role is not clear yet. So dapat wa nila nagipangalanan, no? But then siguro nasa na nakita nga structure. So mind you, the molecular biologists biology is very kanang ano siya, because we cannot see this genetic structures in our naked eye. So very imaginary siya. So we are talking this one, but we cannot see them as a thing, no? But we are just imagining by our minds that they are parts of our constituents, no, or body. So in prokaryotic transcription, prokaryotic cells contain a single type of RNA polymerase, which are in two forms, the core polymerase and the holoenzyme. The core polymerase, they call this, which is capable of RNA elongation but not initiation. The holoenzyme naman is composed of the core enzyme and the sigma factor na required for transcription initiation. So these are just mga RNA polymerase that are needed for the transcription initiation. So this is just to show you the figure what we have discussed earlier in the prokaryotic transcription. So ito yung mga figures niya under a very high-tech na microscope, no? Electron microscope. So holoenzyme and the core enzyme in the prokaryotic RNA polymerase. So a transcriptional unit extends from the promoter to the terminator, and the promoter is composed of a DNA sequence for the binding of RNA polymerase. So the start side, which is the base to be transcribed. And during the elongation, the transcription bubble moves down the DNA template at a rate of 50 nucleotides per second. So medyo madami siya. And transcription bubble consists of actually RNA polymerase, DNA template, and growing RNA transcript. So in structure or in figure, visually, this can be shown on this figure, no? This is the RNA polymerase and the start side na sinasabi kanina. And these are the strands of DNA, yung template. So inside, you can see here unwinding and coding strand, and then rewinding, kaya siya naka-coil, no? Type. And this is the template strand and the transcription bubble. So akala mo kanina yung bubble yung circle, no? But no. This is they call the transcription bubble, then upstream mRNA and the RNA polymerase moving downstream. So ganyan yung picture sa ating this is transcription. And transcription stops when the transcription bubble encounters a terminator sequence. So includes usually 80 base pairs. And in prokaryotes, transcription and translation are often coupled, occurring at the same time. So doble-doble siya at the same time. So this is again another figure to show you RNA polymerase, and when DNA and RNA polymerase dissociates. So may ano siya, parang wire na ano siya, no, mabinat. And this will show you the 4 or more U-ribonucleotides. And this is mRNA hairpin causes RNA polymerase to pose. So etong mga color-coded are actually the 4 bases, no, or the nucleotides. The cytosine is the pink one. The yellow one is the guanine, and the adenine, the violet purple, and the uracil, the bluish one. So hopefully, before you leave this, or before kayo makapasa ni test, you should know that 4 bases of genetic, no, or the central dogma. Etong from DNA magiging RNA, from DNA transcription to RNA, and RNA translated to DNA with the 4 bases or nucleotides. So kahit man lang maalala nyo yan. So I understand kasi even me is kasi sa molecular bias, I have said medyo ano talaga siya, kanang imaginary or imaginative. So again, this is the RNA polymerase, no, showing you the figures. Ito yung under a microscope. So ana lang yun na mo makita sa microscope. But in when you translate, this is under 7.04 microns, no? And under if i-draw mo yan siya, ita-translate mo, parang ang tawag, interpret mo yan as like this. So, ito yung mga mRNA. These are the ribosomes here. And this is the DNA itself, no? Ganyan lang ang makikita mo. But very, if magnify mo pa, yun yung mga coil-coil na. The polyribose and the polypeptide change. So, dito. So, ito yung imagine, no? So, napaka imaginative and creative ng mga molecular biologists talaga. So, eukaryotic transcription will involve RNA polymerase 1, 2, and 3, wherein they transcribe RNA and some snRNA and other small RNAs. And each RNA polymerase recognizes its own promoter. In the eukaryotic transcription naman, initiation ng transcription ng mRNA requires series of transcription factors. So ano ito? These are proteins that act to bind ng mga RNA polymerase to the promoter and initiation transcriptor. So we also have eukaryotic pre-mRNA splicing. So in eukaryotes naman, the primary transcript must be modified by addition ng 5-cap 3 poly A tails. So kanina, as you have seen here, pero makikita nyo, so may mga 3, 5, 5, 3 tails yan, no? And removal of non-coding sequences called introns. So in the splicing, the spliceosome is the organelle responsible for removing introns and splicing exons together. So the snRNPs are small ribonucleoprotein particles within the spliceosome recognize the intron-exon boundaries. So ang Introns are the non-coding sequences, and the exons are the sequences that will be translated. So the tRNA and the ribosomes, the tRNA molecules are the one that carries the amino acids to the ribosome for incorporation into a polypeptide. While the amino acetyl tRNA synthesis are the amino acids. So lahat ng mga aces no, ending in aces are the amino acids yan sa lahat. So acceptor arm ng tRNA. So the anticodon loop contains 3 nucleotides complementary to the mRNA codons. So as shown here in the figure, this is called the 2D cloverleaf model. So kasi ginamit na lang yung cloverleaf genes. So ito yung makikita mo sa acceptor end. Ito, 3 tail and 5 tails. So in the 3D ribbon-like model naman, the acceptor end looks like this one, anticodon loop. And in the 3D space field model, ang acceptor end look like this. And the anticodon here. And so pag maging icon siya, ito yung acceptor end. So nakakatuwa no ang iba't ibang drawings. So here naman are the different steps no into from tRNA going to the charged tRNA traveling to a ribosome. So kanina, I believe we have mentioned this already from the previous discussion. So in figure, ito yung mga nangyayari. As you can see, in the first step, the reaction of the amino acid site is activated and then reacts with ATP to produce an intermediate carboxyl end, ito, of the amino acid attached to the AMP. And the 2 terminal phosphates are cleaved from ATP in this reaction. And then the second step now remains bound to the enzyme. So itong tRNA are bound to the enzymes. Ito yung enzyme or amino acid. Then hanggang sa mag-react na naman, no, mag-translate na ang amino acid from AMP to tRNA. Ayan. Hanggang pupunta na siya sa amino acid attached to the 3 acceptor stem of its RNA. So the tRNA and ribosomes in the ribosome has multiple tRNA binding sites. So we have the P site, the A site, and the E site. So lahat sila will bind to tRNA attached to the growing peptide chain, and in the A side to the amino acid, and E side to another amino acid. So this will show you the figure here. Yeah. So hindi ko na i-discuss kasi in the details. So lahat naman ng explanation nandito sa figure kasi less than a minute na lang tayo. So the ribosome has 2 main functions, no? Decodes the mRNA and also form the peptide band. So ito yung main function ng ribosome. And the peptidyl-transferase is also another, as I have said, lahat ng nag-end ng ACE are enzyme and component din siya ng ribosome which forms peptide between the amino acids. So in the translation, so transcription pa lang yun ha. And in the translation, it requires the formation na ng initiation complex including the initiator tRNA and so on. And binding sequence ng mRNA and its complementary parts. So ito naman yung mga steps na nangyayari sa translation. Ayan. So initiation factor and so on. So initiation complex. So sa initiation stage pa lang ito. So, meron din tayong elongation of translation which involves the addition of amino acids. So, here again to show you the steps, no? From here, and then yung amino acid group will grow into this different amino and terminus.